I welcome you all to my channel. Today I'm going to talk about the triple sugar iron test. This is a test that confuses many students studying the biochemical reactions in microbiology. By the end of the video, hopefully all your doubts with regards to the principle and the interpretation of the test results will be clear. Now, many biochemical tests like the catalase test, coagulase test, oxidase test are carried out for the identification of bacteria in the laboratory. Utilization of sugars plays a crucial part in the identification of gram-negative bacteria, especially the members of the family Enterobacteriaceae. TSI medium is one such medium. Now, the two main purposes of the TSI medium is fermentation of glucose, lactose, and sucrose. And when the bacterium utilizes either of these sugars, whether it is producing carbon dioxide also, along with the acidic metabolites that are produced. The second purpose of this medium is production of H2S when the bacterium is utilizing sugars. So certain bacteria produce H2S on sugar fermentation. Now, before we proceed to understanding the TSI medium, its principle, etc., let's first understand fermentation of sugars. What does it mean? Now, most pathogenic bacteria, rather majority of pathogenic bacteria utilize sugars like glucose, lactose, sucrose, etc. as a source of carbon. Such bacteria are called as saccharolytic. Now, this utilization of sugars by the bacterium can occur in the presence of oxygen or in the absence of oxygen. Utilization of sugars is said to be oxidative when it occurs in the presence of oxygen. So in such conditions, oxygen acts as the terminal electron acceptor. So strict aerobes like Pseudomonas burkholderia, these can utilize sugars only oxidatively. Now, utilization of sugars is said to be fermentative when it occurs in the absence of oxygen. So bacteria that ferment sugars can manage with molecules other than oxygen as the terminal electron acceptors. So now let's consider facultative anaerobes, which are able to grow in the presence of oxygen as well as in the absence of oxygen. So when they are growing in the presence of oxygen, they are utilizing sugars oxidatively, and when they are growing under anaerobic conditions, they are utilizing sugars fermentatively. That means they are fermenting sugars. So members of the family Enterobacteriaceae or Staphylococci or Streptococci, when they are utilizing sugars anaerobically, we will call it as fermentation of sugars, right? So oxidative utilization of sugars occurs in the presence of oxygen, and fermentative utilization of sugars occurs in the absence of oxygen. And what are we checking for today? We're talking about fermentation of sugars. With that done, let's see our triple sugar iron medium. This is what it looks like when it is uninoculated. The blue arrow is indicating the slant, which is the providing the aerobic conditions for growth, and the red arrow is pointing to the butt, that is providing anaerobic conditions for growth. The results of the test are always interpreted and expressed in both the slant and the butt region. So let us start by inoculating the TSI medium. The bacterial colonies are picked up with a sterile straight wire and first stab inoculated right up to the bottom of the butt as shown in figure 1. Then the wire is withdrawn straight back along the same stab line, and then stroke inoculation is done on the slant as shown in figure 2. The medium is then incubated overnight at 37 degrees Celsius. So what are the important constituents of the TSI medium? It contains three sugars, lactose, sucrose, and glucose in a ratio of 10:10:1. These sugars are acting as the source of carbon. Now remember that glucose, being a monosaccharide, is going to be fermented by the bacterium first. So as long as glucose is present, the bacterium is only going to utilize glucose. And whenever sugars are utilized, acidic byproducts are produced with sometimes also carbon dioxide. So if CO2 is produced, we are going to call it as aerogenic. If CO2 is not produced, we are going to call the bacterium as anaerogenic. Now, once the glucose is finished, it's totally consumed in the medium. Now the bacterial cell is going to look for alternative source of carbon. If lactose or sucrose or both of the operons are present, now these will be activated. Bacterium is going to utilize either of these sugars or both of them. Lots of acidic byproducts are going to be produced with or without carbon dioxide. If the bacterium isolate does not have the operons to utilize lactose or sucrose, now it will utilize peptones. And when peptones are utilized, always alkaline byproducts are produced. Remember, peptones are only utilized aerobically. So the result of peptone utilization is going to be only seen along the slant of the TSI medium, right? Now, for detecting the production of acidic byproducts or alkaline byproducts, pH indicator is added, that is phenol red. This is yellow in acidic conditions, which are produced on sugar utilization, and it is red in alkaline conditions, which are produced when peptones start to getting utilized by the bacterium as a carbon source. Now, for detection of H2S production, ferric citrate and sodium thiosulfate are added in this medium. When H2S is produced, these chemicals will combine with H2S to form ferrous sulfide, and this is going to be seen as a black precipitate. Remember one very important point that I had mentioned earlier also, that H2S is formed only when sugars are fermented. That means products of H2S, rather the indicators of H2S, the black precipitation is going to be seen only in the butt of the medium. That is where the anaerobic conditions are present. So now let's see how the results are going to be written. And then we will see with the help of diagrams what are the interpretation going to be. The letter K is used for alkaline reaction. The letter A is used for acidic reaction. G is added if CO2 is produced, and H2S is added if it is seen that there is a black precipitate. So the results are written as the slant reaction and the butt reaction. So slant reaction over the butt reaction, or slant reaction slash the butt reaction. For example, if the result is written as K/A, it means K is indicating the alkaline slant and A is indicating the acidic butt. So meaning peptones have been utilized aerobically and the but is acidic, meaning sugars have been utilized in the but, that is the anaerobic conditions. Now let us see these diagrams or the results and with that we will learn how to interpret the reaction of the bacterial isolate. So here we can see a red slant and no change in the but. So the result will be written as K/NC and interpretation is no fermentation of sugars. Only peptones have been utilized along the slant which are providing an aerobic conditions. An example of a bacterium which will show this kind of result is Pseudomonas. On the left hand side, we can see a red slant and a yellow butt. So the result is going to be expressed as K/A. Since no gas is seen, you can't see any gas bubbles. You can't see a black precipitate. So CO2 as well as H2S have not been produced. So our interpretation is anaerogenic glucose fermentation only. Example is Shigella. Next, we can see on the right hand side an alkaline slant and an acidic butt. CO2 has been produced. You can see that gas bubble right at the bottom which has lifted up the medium. No H2S. The result is going to be written as K, indicating the alkaline slant, slash Ag. So what is our interpretation going to be? Aerogenic glucose fermentation only. So in both these results, you can see that acid has been produced in the butt. Once the glucose got consumed, since the bacterium did not have the lactose or the sucrose operons, it shifted onto utilizing peptones along the agar slant. So alkaline conditions in the slant and acidic conditions in the butt are seen. In these two diagrams, let's concentrate on the left one. We see a red slant and a black butt with gas bubbles here and there. So what is our result going to be? It is K/Ag, K indicating the alkaline slant, Ag indicating the acidic butt, gas indicated by the bubbles, and H2S by the black precipitate. Now you must be wondering, how did I say that there were acidic conditions in the butt? Remember, H2S, which is seen in the form of the black precipitate, is going to be produced only when sugars are fermented. So it's obvious that the yellow color of the acidic conditions have been masked by the black precipitate. So interpretation is aerogenic glucose fermentation only along with H2S production. Remember, the alkaline butt is indicating non-utilization of lactose and sucrose, hence peptones have been utilized aerobically. Example of bacteria which give this kind of result are most serotypes of Salmonella and some strains of Proteus which are aerogenic. On the right hand side, we can see a red slant and a black butt with no evidence of CO2 production. You cannot see any gas bubbles. So what is the result going to be? K/A, H2S. Right? Alkaline slant, acidic butt, no CO2, H2S positive. So our interpretation is anaerogenic glucose fermentation only with H2S. Again, I'm reiterating the alkaline slant is indicating no utilization of lactose or sucrose. Instead, after glucose got over in the medium, peptones started to get utilized under aerobic conditions. Example of such a bacterium which will give this kind of result K oblique A plus H2S is Salmonella typhi. Remember, Salmonella typhi is anaerogenic. Also, Proteus strains, some of them are anaerogenic. Now in this slide, you can see on the left, we see a yellow slant and a yellow butt. So what is this telling us? It is an acidic slant and acidic butt. We do not see any CO2 molecules, neither do you see a black precipitate. So we are going to write the result as A oblique A, and it is going to be interpreted as anaerogenic glucose and lactose and or sucrose fermentation with no H2S production. Okay. Next, on the right hand side, we see a yellow slant and a yellow butt that has been lifted by the CO2 molecules produced on the fermentation of sugars. Right? So acidic slant, acidic butt, gas production, no H2S, result is going to be A oblique AG. Interpretation, aerogenic glucose and lactose or sucrose fermentation or both fermentation. Okay. So example of bacteria which give this kind of result is E. coli and Klebsiella. On this slide, we can see on the left an acidic slant, a black butt indicating H2S production on fermentation of sugars, no CO2 molecules. So our result is going to be written as A oblique A, comma H2S, interpreted as anaerogenic glucose and lactose and or sucrose fermentation with H2S production. Example of a bacterium which shows this kind of result is Citrobacter frondi. On the right hand side, you can see an acidic slant, an acidic butt, CO2 has been produced, and H2S is positive. Again, I'm repeating, why do we say it is an acidic butt? In both these images, you can see H2S has been produced, and H2S is always seen only when sugars have been fermented. So acidic conditions are there in the butt. So result is going to be written as A oblique AG, comma H2S, interpreted as aerogenic glucose and lactose and or sucrose fermentation with H2S. So finally, to summarize the triple sugar iron agar test, if a bacterium only ferments glucose, little amount of acid is produced because of the limited availability of the glucose. So only the butt remains yellow, the slant becomes red because of the switch over of the bacterium to peptone breakdown if lactose or sucrose or both are fermented. Lots of acid will be produced. So the butt will be yellow and the slant also will be yellow. Peptones will not be utilized. Some bacteria may also produce carbon dioxide when they break down sugars. Some bacteria may also produce H2S when they break down sugars, and we've already understood how we are going to detect either of these gas production. If the isolate is a non-fermenter, peptones will be utilized, alkaline conditions will be produced, but these will be seen only along the slant because peptones are utilized only under aerobic conditions, right? And as mentioned earlier, production of H2S is seen in the form of black precipitate in the bud. That's all for now on the TSI medium. Hopefully this will clear your doubts on this medium. See you again with another video to make microbiology easier for us to understand.